Hello everyone, and welcome to our online lecture series for Bio2. Today we continue on with our understanding of DNA control in the nucleus. Thus far we've talked about the process of DNA replication with the different enzymes like topoisomerase, helicase, the single-stranded binding proteins, our DNA polymerase III with those sliding clamps, primases, ligases, and DNA polymerase I, and the directionality of DNA, and how that contributes to the process of being able to copy and make two identical fragments of DNA from a single double helix. We've then also talked about how you can go from DNA to RNA in the process of transcription. We introduced the RNA polymerase molecule, which conveniently has a direction. It's the same as DNA polymerase III. We talked about how DNA copied into RNA is a temporary primary transcript that then can get processed into a spliced final mRNA, and then how those different mRNAs, rRNAs, and tRNAs combine in the process of translation at the ribosome, small and large subunits, in the E, P, and A site in order to create a polypeptide chain or protein. Now altogether, this process is happening constantly in so many of our cells. And the ability to control this process, the ability to control expression, is so incredibly important because a control of gene expression is ultimately a control over metabolism and all of the functions of life. And in today's lecture, we're going to take a look at how gene expression is controlled in prokaryotes and eukaryotes. Now, generally speaking, prokaryotes are much simpler. By not having DNA bound in a nucleus, you have the DNA, the ribosomes, the mRNA, and proteins all within one major bag. So, once the mRNA starts to be made, then you go through the entire process. It is a one-step begin to the end. So we are going to be looking at a lot of regulation early on, so you control what mRNAs are made, and therefore you control expression in prokaryotes, whereas eukaryotes have a large number of different checkpoints. Let's take a look at those checkpoints by re-examining our general process. So, now that we have our red marker, eukaryotic control can happen in a number of different places. First and foremost, it can happen at the DNA because we know that DNA is actually coiled as heterochromatin, so you can control the coiling of DNA in order to control gene expression. Our second checkpoint of control is actually at this RNA polymerase primary transcript step. If we control the creation of a primary transcript, we can control gene expression. Additionally, if we can control the process of splicing, the ability to go from primary to readily read transcript, we can control gene expression. Fourth, if we can control the process of translation starts and the ability to make proteins from your mRNAs, you can also control expression. And fifth and final, what if you can control what happens with the protein afterwards, the endpoints, the destruction of that protein? Well, then you'd also control expression as well. And so generally speaking, looking at this schematic, you can control it expression before anything's made as it's being made into an mRNA, as the mRNA is being processed, as the mRNA is getting ready to go to translation, and then after translation, you can affect the longevity of that protein itself. So we will actually begin our lecture by looking at the more complex version of eukaryotic control, and then return to understand prokaryotic control in a couple of specific cases called operons. So, eukaryotic expression control, as first mentioned, can occur with the DNA and its coiled form. Now, as to whether the DNA is coiled or uncoiled, whether that chromatin is coiled or uncoiled, we call that kind of control epigenetic control. And there are two different types of epi, or on top of, gene control. And this is literally by standing on top of, regulating from above. We can activate or deactivate certain stretches of chromatin through chemical changes called methylation, which affects the state of the DNA, and acetylation, which affects the state of the histones, the proteins on which DNA is wrapped around. When we're talking about methylation, we are often talking about regions that have been blocked off, regions that we want to keep silent, such as heterochromatin. This is often done in a CG context, meaning that where we find a CG, we'll oftentimes put a particular chemical modification that we as scientists indicate with a CPG, preferentially in mammals. Plants have 3 different types of epigenetic control, so we'll stay out of that. And then we look at how that gene is either turned on or turned off in different lineages. And we can see these differentially methylated regions, or DMRs, as oftentimes playing a large role in how phenotype manifests in different looking organisms. So the genes, even though the DNA might be the same, the methylation context of those genes in different individuals might be different, therefore causing the phenotypic differences. Now functionally, this looks like this, where you have genes that are wrapped up and the DNA is inaccessible because methyl groups are causing the coiled chromatin to hug closely to itself. So methylation generally will cause a close of the DNA. In some cases, to undo the closing that occurs with methylation, you can impact the way the histones are used to wrap the DNA around them. You can create more space between those histones. The space between the histones can be increased by adding an acetyl. So an acetylation will generally cause the opening of DNA. When DNA is open, this allows for RNA polymerase to come in and begin the reading. When DNA is closed, RNA polymerase can't make it in. So epigenetic control is that first, before you begin, before a transcript is made, form of control. One of the most common places that we see this epigenetic control is in the Barr body. Now the Barr body happens to be the specific name of one of the 2 female X chromosomes. Now you'll notice females in humans are XX, males in humans are XY. Now we've talked a lot about gene dosage and how you're going to want to get the correct number of chromosomes in an organism in order to not have things go poorly. So how does this work in humans, where a male has 1X and a female has 2X, but the dosage would be different in these two different groups, but neither looks severely developmentally challenged? Well, in this case, with female XX, one of those chromosomes entirely is methylated, causing it to essentially be silent, for it to not be there in each individual cell line. This methylation causes the chromosome to go from being a giant X and open and red to being a tiny, tight little ball of heterochromatin, all wrapped up, much smaller than it would be normally. And we call that small wrapped up methylated chromosome a Barr body. Now, Barr bodies are super interesting because they're actually responsible for the color pattern in things like calico cats. You'll notice that it is much more common for a female cat to be a calico than a male. And this is because female calico cats carry 2 X chromosomes, and in different parts of their body, one or the other X chromosome is turned off. By turning off the chromosome that carries a pigmentation gene, so making color in the cat, you will get a patch of calico cat that is white. And by turning off the X chromosome that carries the lack of pigment, or the white coloring, you will get a patch of calico cat that either expresses the black or the brownish fur. And in this way, you can get a cat that has essentially 2 different pigmentation pathways based on methylation in different groups of cells across their body. You can literally visualize this DMR, differentially methylated region, across the cat's body by looking at where color is and is not being expressed, or at least is being expressed differently. For our second form of control, we have to look at the control of the RNA polymerase, which we'll call transcriptional control, or control that occurs at transcription. Here's the image corresponding from your book about transcriptional control. Now, I won't expect y'all to keep track of all of these, but know that there are elements that at transcription that can cause DNA to bend, that can enhance, that can activate, and that can block. These different elements that are far away, distal controllers, are oftentimes enhancers. They find ways to make the expression of this gene higher based on what's going on super duper far away. The stuff close, the proximal controller, would be something called a transcription factor, a protein that turns transcription up or down based on its affinity to a DNA sequence called the promoter. An upstream fragment of DNA, so it's something that hangs out before the gene, that serves as a way to recruit RNA polymerase and its other associated components. So in terms of transcriptional control, we have those things that act far away, are enhancers, and we have those things that act close up, like TFs, or transcription factors. Once you've created a functional transcript, Once you've made that RNA, then you can look at RNA processing or post-transcriptional control. And in post-transcriptional control, you're going to begin with your pre-mRNA. Now, the thing about pre-mRNA is it can actually end up being different products after splicing. What exactly do I mean by this? Well, if you have your pre-mRNA with 4 exons, exons 1, 2, 3, and 4, in one spliced product, you might find exons 1, 2, and 3. In another spliced product, you might find exons 1, 3, and 4. These two different splice versions are called gene isoforms. When one primary transcript can result in several or even many, there are currently, in some cases, more than 10 possible isoforms noted from genes that have many different exons, you can get this kind of post-transcriptional control that can tweak gene versions to one isoform or another. Additionally, there's another way of controlling mRNA expression based on the fact that these spliced mRNAs are single-stranded. Broadly speaking, this type of control relies on what is called RNA interference, or RNAi. RNAi can be accomplished with either short interfering RNAs, siRNAs, short RNAs, or microRNAs. Not that y'all need to keep track of any of these in particular, but just know that they have different mechanisms, slightly, to have essentially the same product of interference. So what happens with these interfering RNAs is that you're going to get a single-stranded spliced mRNA that finds its exact matching product of another single-stranded RNA and binds to it. So you complement that single-stranded RNA. By complementing it and now having two RNAs bind it together, this renders the RNA null and void. Can't break them apart. It's got to now be broken down. It gets broken down by something called the risk factor, or the RNA-induced silencing complex. And the risk factor, this complex that comes and binds these double-stranded RNAs, goes ahead and recruits dicer, which is not some sort of acronym, but named because it finds double-stranded RNAs and chops them up. Whenever they're bound to one another, it dices them up. When the spliced mRNA is diced up, it's no longer going to be expressed. And so in that way, eukaryotes can control what happens with their spliced mRNAs after they've already been spliced, in case you need to change the signal. Lots of different control, right? Coiling beforehand, recruitment of the polymerase at the site of transcription, post-transcriptional control with different isoforms and even breaking it down as it's on its way to its ribosome. So next up, we'll talk about the type of control that occurs at translation, or translational control. From our diagram here, we're talking about the stuff occurring here at step 4, translational control. In translational control, we are essentially going to want to regulate initiation. That initiation part of translation. Now this is done through a series of factors and phosphorylations. So let's go ahead and take a look at this image, figure 16.3 from your textbook. Now what this image is showing are some modifications that can occur to the small subunit of the ribosome to either progress or stop translation from occurring in the first place. And the way this works is that the small subunit of the ribosome is going to have to interact with the EIF2 protein. The EIF2 is the eukaryotic initiation factor. -2, because there are other initiation factors. And it's the eukaryotic one because it's actually conserved across the vast majority of eukaryotes. So me and my dog happen to have the exact same sequence for this eukaryotic initiation factor on the protein level. Kind of cool. Well, the eukaryotic initiation factor interacts with an energy molecule called a GTP, or a guanosine triphosphate, an analog to ATP that has many roles inside of the body, inside of cells generally, to control different functions. When EIF2 binds a GTP, it becomes phosphorylated, therefore preventing any of the large subunit or mRNAs to come in and start translation. When instead EIF2 is not phosphorylated, translation can occur. And so in this way, by controlling the GTP levels in your cell and how the GTP interacts with EIF, you can globally shut down temporarily translation. This has a lot of potential different roles, particularly in things like infections, where if you want to temporarily shut down translation globally because you happen to not be translating your own proteins, but instead those of a virus, cells can do so with these global translational kill switches. Next up on our exploration of expressional control is our last step, the step when the protein is made and floating out in the eukaryote. Therefore, we call this post-translational control. Generally speaking, post-translational control operates on either a reversible mechanism, where you add some sort of functional group to the protein. Oftentimes, this is actually a phosphate group, similar to what we see here in the EIF2, whether it's phosphorylated or not. Changing the chemistry of the protein can temporarily change whether it's translating or not. Or the irreversible. And that is a process called proteolysis, or protein destruction. The way that this works is that you've identified a protein that no longer needs to do its job. Perhaps the threat of the tiger behind you is gone, and your body needs to now ramp things down so that way you can catch your breath. What would happen in this case is the protein that's no longer needed would be treated with a chemical marker called ubiquitination. Ubiquitination would then cause the recruitment of a proteasome. And that proteasome would start to break down your giant protein into its monomer amino acid constituents to allow those amino acids to be picked up by different tRNAs and then reincorporated into a different protein. So basically, the irreversible proteolysis is protein recycling. And these are the 5 major ways in which we can control gene expression in prokaryotes. In eukaryotes, not prokaryotes. We can control it at the DNA level, whether the DNA can be read. We can control whether that DNA is being read quickly or slowly by RNA polymerase at the transcriptional level. We can control the post-transcriptional processing, either in different gene isoforms or RNAi. Translational can control where we want to either temporarily shut down or ramp up processes of translation across the cell with methods like the eukaryotic initiation factor 2 and GTP. And last but not least, our method of reversibly, post-translationally adjusting protein function or recycling proteins in an irreversible proteolysis reaction headed by ubiquitination. Those 5 major categories additionally have subcategories that we won't dive into, and instead we'll end our discussion of eukaryotic expression control here and now switch to prokaryotic expression control. Which is at the same time much more simple in that it occurs at only one step, but also elegantly cool in how well this regulates the metabolism of these tiny bacteria, these tiny prokaryotes. The general idea behind prokaryotic expression control is to prevent or stop transcription. In eukaryotes, you can oftentimes waste a lot of metabolic function in controlling epigenetics, in controlling transcript production, in controlling post-transcript production, or even getting down to the either translational or post-translational control. You can imagine how satisfied a factory worker, the ribosome, might feel if the transcript is made, the transcript is processed, the protein is made, and then immediately after the protein is made, the body sends a signal to break that protein down. Now that might serve the long-term survival of the organism, but metabolically, that's a lot of expense. Eukaryotes don't always have such a emphasis on making sure that they are optimized for, or rather, overall they are optimized for good use of energy, but they can absorb a bit more of metabolic waste because things are not, because they have to find ways to create more energy than they will use. Prokaryotes are oftentimes much more concerned with controlling the metabolism before anything happens. There's no reversible process here. You want to stop transcription before it begins in order to help this prokaryote operate and survive. This prevention of transcription occurs with the use of a prokaryotic operon. An operon is defined as clusters of genes that will be turned on or off at the same time. When an operon or operator is turned off, we term that as a repressor operon. And when it is turned on, or the thing that is turning the operon on, we call it an activator. So you have things that turn operons off, repressors, and things that turn operons on, activators. So we're going to look at this process of operon control in two specific examples. The first in the tryptophan operon, or the trip operon. Tryptophan being an essential amino acid that's used in many different protein synthesis as well as other functions within the organism. And this trip operon is known as a repressible operon. Let's go ahead and draw out the trip operon in this space here. The most intuitive part of the trip operon, abbreviated as TRP operon, so that we keep track of it over here, are what are called the structural genes. These are a cluster of 5 genes: trip E, trip D, trip C, trip B, and trip A. That are 5 genes literally smashed adjacent to one another. These 5 genes code the 5 different proteins that are necessary to go from the metabolic precursor to trip to the end product of actually being trip. So from start to finish, they make the amino acid tryptophan. So they really are the product that we're trying to regulate. Does this bacteria currently need to make tryptophan? If yes, make these 5 structural genes. If no, then don't. The regulatory components of this are the DNA sequence upstream. So now we need to clarify what upstream and downstream means. So upstream are things in front of, downstream are things behind. And that'll matter in a big way in just a second. So upstream of the structural genes, you have the DNA sequence that is controlling their transcription. This is called the operator. It's very important to know that the operator is DNA sequence. The operator will bind to a molecule. The molecule it binds to is called a repressor. In this case, the repressor is a protein. Upstream of the operator, we have our final DNA component, that being the promoter. The promoter is also DNA. So, how does this work? Well, the repressor will stay bound to the operator as long as the cell has enough tryptophan. If the cell doesn't have enough tryptophan, the repressor falls off. So with enough tryptophan, the repressor is staying and blocking. Well, how does that prevent transcription? Well, RNA polymerase is going to bind at the promoter. So let me go ahead and label this. Now, if RNA polymerase binds at the promoter, which it's trying to do frequently, and it tries to go past the operator, starts to perform transcription, it's going to bump into the repressor and fall off. Come back on, bump, fall off. Come back on, bump, and fall off. So as long as the repressor is there, RNA polymerase won't be able to do anything. If we can peel the repressor off, in circumstances where the cell needs tryptophan, well then the RNA polymerase will read through, make EDCBA, fall off, come back around, make EDCBA, and fall off. And in this way, you're actually able to make tryptophan in cases where the repressor is not present. So the missing component to this entire diagram is what pulls the repressor on and off. In cases that the repressor is bound, the cell actually has enough tryptophan to spare it to bind to the repressor. And when tryptophan is present, the repressor is of a conformation that will bind the operator. If the tryptophan gets pulled off and used for some other metabolic function, then the repressor changes conformation, will peel off the operator, and transcription can begin. If tryptophan was to be made in high amounts, it will then bind to the repressor again. Once bound to the repressor, it will then look for the operator and again bind the operator. In this way, when TRP levels are high, don't make more, is what the cell is doing. When TRP levels are low, transcribe the structural genes, or SGs. I'll abbreviate them short here. So your book also has this diagram essentially illustrating the same premise. See if we can get rid of that thing there. When tryptophan is present, the TRP repressor, a protein, will bind the operator and RNA synthesis is blocked. The polymerase can bind at the promoter, but it's not going to go past the operator. The repressor is sitting in its way. When TRP is low, you get that conformational change in the repressor. It looks different. When it looks different, it cannot bind at the operator. And in that case, it is disassociated from the operator and RNA synthesis will proceed. You'll go ahead and make the genes that will go ahead and make you more tryptophan. When you've made enough tryptophan, you then control the repressor through negative feedback to go and tell the cell to stop making more of it. So when you have enough, the bacteria doesn't make it. When you need some, it will make it. And it does so by controlling the repressor, a protein sequence, and by having specific players interact with the operator, the repressor, and the promoter, the RNA polymerase, constantly. Because you are dealing with this negative regulation coming back, we generally call the TRP operator a negative operon. And the only other detail to make it completely accurate is that it's not just a single tryptophan, but you need two tryptophan molecules to bind to the repressor in order to actually peel it off. So 2 trips. A lot of tryptophan present will be able to peel the repressor off. If the tryptophan is needed for something else, it gets peeled off of the repressor, and then the repressor will no longer bind to the operator. Fairly clever, right? At least I think so. With this operon under control, let's go ahead and take a look at our next operon, the lac operon. This time, instead of being a amino acid like tryptophan, lac codes for the lac operon codes for the proteins necessary to turn lactose, a sugar molecule, into readily available energy for the cell. The lac operon is an inducible operon, meaning that you can turn it on when necessary, because you don't always want it on. How often do you think a prokaryote finds itself in an environment where lactose is the readily available sugar? Well, lactose is the sugar produced by mammary glands in milk. That means that this bacteria would need to find itself in a giant vat of milk in order to want to break down lactose for sugar. Because lactose is so scarce, you are only going to want to turn the lac operon on in cases when you have low glucose, because glucose is the preferred sugar. It's much more available. It's produced by plants, and there are a lot more plants on this planet, a lot more green things than there are milk-producing things on the planet. So you're going to want to turn the lac operon when glucose is low, and lactose is actually present. These two describe the on condition. Everything else, generally speaking, should find the lac operon off. So, how does this work? Well, just like in our trip operon, you have some structural genes, 3 of them in this case. Those genes being the LAC-Z, LAC-Y, and LAC-A. Our 3 structural genes. Upstream of our structural gene, make sure I follow the color patterns here. We have our operator, our giant stop sign, or start sign, go sign rather, and then an upstream promoter. And just like before, the operator is DNA sequence, the promoter is DNA sequence. We also have a repressor, which is again protein sequence. We again have our RNA polymerase molecule. But there's one other component, one additional thing. The lowercase c, capital AMP complex. A CAP-CAMP complex that's going to play its role with the RNA polymerase. So what is CAP-CAMP? We know that the promoter is DNA, the operator is DNA, the repressor is a protein. The CAP-CAMP complex is a catabolite activator protein. So it is a protein sequence. This protein sequence is going to be found bound to the promoter of the lac operon. And it's found bound to the promoter when it is in this CAP-CAMP form. It can alternatively be changed into just a CAP form. The CAP is actually this catabolite activator protein. The CAMP is known as cyclic AMP, or adenosine monophosphate. AMP is the depleted form of ATP, which is our cellular energy. So when the cell runs out of energy, it goes from ATP to ADP to AMP and then cyclic AMP, signaling that the cell has low energy. We're starving. Get us some food. Cyclic AMP will then bind with CAP to form the catabolite activator protein in its inducible or on form. When CAP-CAMP binds to the promoter sequence, it is going to serve as a recruiter, a stronger recruiter for RNA polymerase. Because CAP-CAMP signals, we're starving, cell. You've got to get us some food and fast. So I'll move this down a little bit here. Now, just like with our tryptophan situation, you actually need 2 times cyclic AMP to bind with CAP in order to make CAP-CAMP. So 2 molecules here as well. Just coincidence, no particular reason behind that. And cyclic AMP will be high when glucose is low, because glucose is that preferred energy form. So when you have low glucose, you are actually having a lot of CAP-CAMP, telling your cell, we're desperate for energy. Please, 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 let's go ahead and make some from anything that you can find. At the same time that CAP-CAMP is doing that, we also have our regulator of the repressor. Now the repressor will be bound to the operator normally. The thing that will pull the repressor off is the presence of lactose. When lactose is there, it'll bind to the repressor and pull it off. In this way, the presence of lactose controls the repressor. When lactose is present, or the presence of lactose, will pull the repressor off the operator, signaling that, hey, guess what? You can go ahead and make this series of lac operon structural genes if you need them. So this is 2-step regulation. Normally, repressor is hanging out, and RNA polymerase is not really being recruited to the promoter that much. Occasionally it'll go through, but if the repressor is there, it'll fall off just like before. The RNA polymerase will put an emphasis on binding to the promoter when energy overall is low and CAP-CAMP is present. But when CAP-CAMP is present and it gets recruited to the promoter, if lactose isn't present, the cell definitely doesn't want to waste energy and the operator is still blocked. The RNA polymerase can be recruited as much as it wants to the promoter, but it will always fall off. If lactose is present and energy is generally low, then the cell needs to communicate, guess what? We can get energy from lactose. We need energy. Go ahead and make it. And so in this case, you need 2 conditions, just like we talked about before. Low glucose with CAP-CAMP, and that alternate energy source actually available. And that's what your 2 diagrams from your book are showing. First, it states that in order for the cell to even explore making the lac structural genes, lactose has to be present. So these two models assume lactose present. If the cell doesn't need energy because also a whole bunch of glucose is present, cap will be by itself, no cap-camp, and even if RNA polymerase occasionally gets to bind to the promoter, transcription will only occur at a low rate. If instead camp is bound to cap, you then have this really good recruitment. And remember, it's 2 times camp per cap. You have this really good recruitment at the promoter, so that way the RNA polymerase is starting to make a whole bunch of lac structural genes in order to harness that energy present in the lactose. So you essentially have 4 situations. And these are something that I expect you to be able to walk me through based on the logic of the environment of the cell. What does the operon look like in each of these 4 different cases? First, if lactose is absent, the repressor binds the operator and transcription is blocked. We don't care what's happening with cap-camp. No expression is happening. In the presence of lactose is there, the lac repressor is released from the operator and transcription may proceed at a slow rate because lactose is an alternate energy source. So therefore the operator looks like this. The repressor pulled off by the lactose, and the RNA polymerase occasionally getting through. This has, again, that slow rate versus transcription fully being blocked. In our third situation, cap-camp stimulates RNA polymerase activity and increases RNA synthesis. This leaves out the fact that lactose is present. Because that operator does not have a repressor bound to it. So when energy is low through that present of the camp with the cap, you would get massive production of the lac structural genes, and this is the case where you're actually fully utilizing the lactose to derive energy from that for that prokaryote. In our fourth case, you need the energy even in the presence of cap-camp, cell is signaling I need energy. RNA synthesis will be blocked if the repressor is bound to the operator rather than to lactose. And so this is what it looks like. Even if the cell desperately needs energy, as indicated by cap-camp, the repressor is going to keep things bound up until it finds its good energy source with lactose. If you don't have lactose, don't make the structural genes. Conserve your energy. Things are getting tough. So what do I expect from you in terms of the understanding of these operons? Well, there's a high probability that you may be asked what is happening with an operon given certain circumstances in the cell. So you should be able to tell me where the promoter, or what is bound to the promoter, what is bound to the operator, where is the repressor bound, what is it bound to, and what is cap-camp doing? Are we in a situation where the cell is full of ATPs, or are we in a situation where the cell might be low on energy and full of CAMP, camps? In the same way, I'd expect for you to be able to explain to me when tryptophan is created and under what conditions the trip operon is operating. Particularly, be able to explain how the negative feedback loop works here. And then a broad understanding of the 5 different places in which eukaryotic expression control can occur. Now, this whole lecture serves as hopefully a brief overview of how we as scientists can begin to pull these processes apart, design new machines, tinker with the genetics, tinker with the proteins in order to A, understand their function, and B, expand on their utility. A lot of the field of bioengineering utilizes a lot of these underlying genetics for human purposes. So know that you're learning these things not just to have to memorize them, but so that way you can think about how you might manipulate them in future work. Kind of cool, huh?